Again, good morning, good afternoon, wherever you're joining us from. Welcome to all of our audience members to today's edition of Sino Biological's Lock and Key webinar series. Our guest today is Spatomics, an ultra-high-plex spatial omics outfit headquartered in Guilford, Connecticut. Before I go on and introduce Spatomics fully, I would like to point out a few housekeeping items. Firstly, please feel free to type any questions into the chat box throughout the presentation. We will try to address as many questions as possible immediately following the presentation during the Q&A session. Secondly, please let us know where you are joining us from today. It's always a pleasure to see what countries are represented by our audience members. And lastly, this presentation will be recorded and uploaded later to YouTube, where you'll be able to rewatch and share. And now, Spatomics. To enable highly multiplexed single-cell in situ analysis, Spatomics has developed cleavable fluorescent probes, CFP, for comprehensive molecular profiling in single cells in situ. In this method, affinity probes, which can target biomolecules with high efficiency and specificity, are conjugated to fluorophores through a chemically cleavable linker. Spatomics offers unique value proposition with its patented efficient CFP chemistry. The innovative system generates comprehensive contextual data, revealing detailed molecular interactions at the subcellular level, while preserving the integrity of the tissue samples. And now, it is my pleasure to introduce our two speakers representing Spatomics. Starting with Xiaoshan Wang, who is the VP of Business Development at Spatomics, headquartered in Guilford. Spatomics aims to build the world's leading intelligent tool platform for spatial biology. Before joining Spatomics, he worked at Advanced Cell Diagnostics, a biotechnique company where he focused on RNA-scope applications. With over 15 years in the biotechnology/clinical diagnostics field, Xiaoshan received his Master's of Science degree in Biomedical Sciences from Virginia Tech and his MBA from the University of California, Berkeley Haas School of Business. Our second speaker today is Dr. Jia Guo, who received his BS degree in chemistry from the University of Science and Technology of China and his PhD degree in chemistry from Columbia University. After doing postdoctoral research at the Stanford University, he joined the faculty at Arizona State University, where he is currently Associate Professor of Chemistry and Biochemistry. His research focuses on the development of ultra-sensitive single-cell in situ genomics and proteomics technologies with novel fluorescent probes, which will enable the systematic analysis of the identities, positions, and abundances of a large number of different biological molecules in individual cells of intact tissues. And without further ado, I will let Jia and Xiaoshan dive into today's talk, titled Single-Cell In Situ Genomics and Proteomics Analysis with Cleavable Fluorescent Probes. At this point, please feel free to share your screen and take it away when you are ready. Thank you.
All right. Thank you, Max. I really appreciate the introduction. And it's our great pleasure to meet everybody here and on this Sino Biological platform. So before we dive into Dr. Guo's talk, I want to give a quick introduction about Spatomics. Let me share my screen. Give me a sec. Here we go. Can you see my screen? Okay. All right.
Yes.
Okay. All right. So Spatomics is a startup company founded in 2023. As our host just mentioned, we are headquartered in Guilford, Connecticut. And we aim to build the world's leading intelligence life science tool platform. And currently, we're focusing on multiplexed spatial platform and reagent using our proprietary technologies. So before we dive into our technology, I want to give a quick introduction about spatial biology as a whole. So I think everybody probably have already used one of the tools either in the bulk or single cell or spatial biology field. So the bulk analysis tools have provided a good insight into the general trend of a gene or a protein expressions such as PCR, DNA sequencing, ELISA. And with the technology advancement, the single cell method allows us to get deeper understanding about the cell types, uncover the cellular heterogeneity, identify the rare cell types and the states. However, both methods, they lose that one important information. The one piece of important information is the spatial information. During either the homogenization process or a cell dissociation process. So beyond the bulk or single cell analysis, spatial biology, on the other hand, they study the location and the organization of molecules within the tissue and organs provide a rich morphological context that the bulk or single cell method miss. So in spatial biology studies, the morphological information is well maintained. It reveals how the cells interact with each other in the microenvironment and how the interactions actually influence the function and disease. So in the spatial biology, there are two main focus apart. One is a spatial transcriptomics, the other one is a spatial proteomics. So both methods interrogate different molecules, either the RNA transcripts or proteins, and together they offer the deeper biological insights. From the technology maturity point of view, I believe the spatial transcriptomics is more mature with a well-established platform such as the 10x Genomics Visium or Xenium platform. Well, on the other hand, on the protein side, there are rapidly evolving new technologies, and today we'll be talking about one from Spatomics. So before we let Dr. Guo to start off in-depth talk, I want to briefly go over how the spatomics technology works. So the spatomics technology actually has been going into multiple iterations already. It is mainly we call it cleavable fluorescent probe, or CFP. It is actually based on the cleavable teramide fluorophores. The technology is developed in Dr. Guo's lab, and we are able to use this technology onto the Spatomics platform. The technology in this first generation is using a antibody direct labeled fluorophores, and in between the antibody and the fluorophore, the Dr. Guo has introduced a cleavable linker, which is the core to the technology. The cleavable linker can be cleaved by a cleavage reagent. So after the cleavage was done, then you could go ahead and then interrogate another protein marker, and we could do that in a cyclic fashion to interrogate multiple protein markers on the same tissue section. However, the downside of this first-gen technology is also very obvious. There's no efficient signal amplification. So for some lower abundance of protein, you will have some sensitivity limitations on that. Although this is similar to some other on-market spatial biology companies offering. So in the second gen of technology, Dr. Guo introduced the teramide signal application that combined the efficient TSA signal application into its technology. So in this Gen 2 technology, the cleavable linker was designed into the fluorophore and the teramide molecule. So in between the teramide and this fluorophore, the linker was built in between. So once the teramide deposited into the tissue, the cleavable reagent still could release the fluorophore from the tissue. In the traditional teramide method, TSA-based method, once the fluorophore is deposited into the tissue, it's permanent. You cannot remove the fluorophore. So in order to interrogate multiple samples, you have to use the different colors of a TSA fluorophore and deposit the fluorophore one by one onto the tissue and image at the same time. So the problem is that once you deposit more and more fluorophores, the spectrum becomes so crowded that then you have to use the high-end spectral unmixing capable fluorescent scanner in order to separate those fluorophores from the crowded spectrum. However, with our technology, you could do in the cyclic fashion. In each round, you only need to image 3 to 4 fluorophores, and afterwards you can cleave off the fluorophore, the targets. So after each round, you can cleave off the fluorophore and start a new round, interrogate more markers. And because the chemistry used for cleavage is very mild, then you could run the cyclic fashion in so many rounds and still maintain the antigenicity and also the tissue morphology. Then I want to share some of our recent work developed in-house. Then the cleavage fluorescent probe is our exclusive chemistry. We have a global rights to this chemistry. And with this chemistry, you only need to use the off-the-shelf antibody. No oligo conjugation needed. No need to do any antibody modification. Take the on-market antibody, and if the antibody works for IHC, for immunohistochemistry, the most likely will work with our chemistry. And because we leverage the efficient TSA for signal amplification, you are able to use this technology to detect the low abundance of proteins. And with other features of the CFP, you can unlock the potential of high multiplexing protein interrogation. And also, since we use the off-the-shelf antibody, which makes the panel designing a lot easier than some traditional method. And currently, we're also developing our own automated spatial platform. Then in later this year, we are aiming to introduce the beta testing unit, so to help streamline the whole workflow. Here, I want to demonstrate some of the results from the lab. Here is the FFP human normal tonsil tissue stained with the CFP. With this staining, we actually only use the one fluorophore. So only use the one fluorophore, we can repeatedly stain, cleave, restain. And in our lab, we have already demonstrated 15 rounds of staining and the cleavage cycle. So you can imagine that if we're introducing more fluorophores, for example, like 3 to 4, we can achieve really high multiplexing in protein detection. As far as I know, currently on the market, there's no other TSA-based platform can image beyond 8 to 9 plex. And with this fluorescent imaging, you could easily interrogate the different markers in different fluorescent channels or different imaging round, and you could also overlay them together for your downstream analysis. I mentioned that our company currently is focusing on high multiplex protein detection. But in the meantime, we're still working with the industry-leading RNA-ish providers and work with them to offer RNA-ish assays using the fluorescent probe. Here is the HeLa cell palette stained with the PPIB RNA-ish probe. And here, this one is the TBP probe stained, also using the RNA-ish probe stained on the HeLa cell palette. So the TBP gene actually is a low expression gene. In HeLa cell, the average transcripts is below 5. So they demonstrate the sensitivity of the RNA assay, and then also the capability of the CFP working on those low expressing genes. The third one is it's a HeLa cell palette stained with Malta-1 probe, which is a long non-coding RNA. So in your study, when it comes to the situation that you don't have a good antibody, then you could still offer to use the RNA-ish probe. And the last one is the negative control. And the images below here also demonstrated the RNA-ish performance on the FFP tonsil tissue we stained previously. And here the antibodies we use to stain those tonsil tissues are mainly from Sino biological. And the last one I wanted to show you guys here is the important work is the RNA protein co-detection assay. And according to the DASI biological market report, the co-detection market share will reach 46% by the year 2029. That is to say, almost half of the spatial biology applications would be in the multi-omics fashion. Spatomics here aims to become an important player in the protein and RNA co-detection market. On the images here on the left, I'm sorry, on the right. So we demonstrated one RNA marker, the PPIB, combined with multiple protein markers. Here is alpha SMA, CD3, HLA-DR, and also Ki67. And the majority of those antibodies are also coming from Sino biological. So here's a quick introduction about our company, our technology. And now I'll pass the floor to Dr. Guo to share more in-depth study from his lab. Thank you, everybody.
Thanks, Xiaoshan, for the very nice introduction. So I think everybody already knows like Spatomics is quite powerful to generating all of these amazing results. So now I may take the opportunities to further introduce like how do we generate those great results in more depth. And also I would like to thank Sino Biological and Max to inviting us to give this to have this opportunity to introduce our company and our research. Okay, let me share my slides. Okay. So our lab is focused on to develop novel technologies for single cell in situ genomics and proteomics analysis. So we are using novel fluorescent probes to achieve that. Okay. Maybe use another pointer, laser pointer. Okay. So first a disclaimer. So I'm a co-founder of the company Spatomics. For all the technologies we are going to introduce today, all of the technologies has been licensed exclusively to Spatomics. And I'm also an inventor for a patent license to a NanoString. So later NanoString developed that technology into a CosMx technology. So today I may not have time to introduce that technology. So again, all the methods I'm going to introduce today is exclusively licensed to Spatomics. So as Xiaoshan introduced, like so for biological analysis, we go through the process like a box cell analysis, then later single cells, then the later spatial analysis. So in 2009, so the journal Nature Methods names the box cell sequencing, the bulk RNA or DNA sequencing technologies, the method of the year. So basically, box cell sequencing is like you're getting a smoothie. So you are mixing like many different types of fruits. You get the average results from a gene expression or DNA sequence information from many different samples, many different cells from the same sample. So you are averaging the results. You're getting a smoothie. Later, the journal Nature Methods names single cell genomics the method of the year in 2013. So within these new technologies, by isolating the individual cells from a complex biological systems, such as brain tissues or tumor tissues, developing embryos, etc. So people can isolate individual cells, then do single cell sequencing. So in this way, we know how many different cell types and subtypes are existing in the biological systems. So we know what they are, and but we don't know their interactions, who is interacting with whom. So for example, to study like memory, we have to know like the interactions between the presynaptic cells, talk with the postsynaptic neurons. So we want to know the biological informations in situ, in their original locations. Or another example is like to study immunotherapies. So we want to know like the infiltrated immune cells, are they really targeting the specific cancer cells? Which immune cells have a better penetration efficiencies? Which cancer subtypes they are really targeting? So we need to have the spatial information to really see these different cell types together, or see the biomolecules in their original cell locations. Then we can have better understanding of the biological systems. So that's why like new spatial technologies has been developed. And in 2020, again, the journal Nature Methods, named spatial transcriptomics, the method of the year. So in this way, we are kind of have a fruit tart. So we really know like individual cells, who is next to whom, how do they interact with each other, what is the microenvironment of the tumor cells, etc. So our lab is in the field of spatial transcriptomics and the proteomics field. So basically, we are developing the highly multiplexed single cell in situ analysis technologies. In other words, we want to quantify the identities, positions, and abundances of a large number of different DNA, RNA, and the protein molecules in individual cells of the intact tissues in situ in their original locations. So if that is achieved, then we can analyze brain tissues to study how does the brain cell functions, how do we achieve these highly complex biological tasks, or in the solid tumors, how does the tumor progress, and finally like metastasize. So what's the molecular mechanisms of the disease? And in developing embryos, we can study how does the single cell become 2, become 4, and later how does the organ is formed. And also during this process, we may also find some new biomarkers for diagnosis or treatment monitoring, or we may also find some new drug targets for more effective cellular targeted therapies. So basically, this is our goal. How are we going to achieve that? So as Xiaozhan mentioned, our first version of the technology, we are conjugating the fluorophores directly to the antibodies or later for oligonucleotides for RNA analysis through a chemically cleavable linker. So if you are familiar with the conventional immunofluorescence assays, basically they are using fluorescent antibodies to recognize their targets. Then after staining under the fluorescence microscope, they can visualize maybe 4 or 5 different proteins under different fluorescence channels. So based on the fluorescence emissions and their intensities, they can see or distinguish 4 or 5 different protein expression levels in their original cellular locations. So with our technologies, we can insert a small chemically cleavable linker between the fluorophores and the antibodies. So after the first cycle of staining, we can chemically cleave the fluorophores and wash away all the cleaved fluorophores. So all the fluorescent signals generated in the first cycle is gone. Then we can apply a second set of antibodies, but labeled with the same set of fluorophores to stain other proteins in the same cells. So in this way, through cycles of staining, imaging, cleavage, staining, imaging, cleavage, they can quantify a large number of different proteins in the same specimen. So to make this technology work, there are 2 requirements. So the first requirement is like we have to do these organic reactions within the cellular environment, and it has to have very high efficiencies. So after the cleavage reactions, after these organic reactions, almost all the fluorescent signals should be gone, so they cannot interfere with the subsequent cycles. And the second requirement for the success of this technology is like these chemical reactions should not damage the epitopes or RNA molecules in the tissues, in the cell. So these intact biomolecules can be analyzed in the subsequent cycles. So to demonstrate our achievement to fulfill the first requirement, so we stain different proteins in the HeLa cells with different fluorophore conjugated antibodies using our cleavable linker. So after the fluorescent staining, after incubating the cells with our cleavage solutions, within 30 minutes, almost all the fluorescent signal is gone. So the red line here represents the fluorescent signals in the stained cells in the red line positions. And after cleavage, the red line, the green line here represents the fluorescence intensities in the green line regions after signal removal. As we can see, even within 30 minutes, almost all the fluorescent signal is gone. And we have to demonstrate like these signal cleavage reactions, these organic reactions that damage the epitopes in the cells. So to achieve that, to demonstrate that, so we first, this is the conventional immunofluorescence shown in panel J. And we also incubate the HeLa cells with our cleavage reactions for 24 hours before we do this staining. So our one cleavage reactions, we can demonstrate, we can do the cleavage within 30 minutes. Then later, Spatomics further optimize that. We can further reduce that to within a couple of minutes. However, so within one cycle, at most within 30 minutes, we can cleave all the signals. But here we incubate the cells for 24 hours. So we are pretending we are doing cleavage reactions for 48 cycles. Then after that, we stain the cells with our fluorescent antibodies, as we can see in terms of staining patterns or staining intensities. So the staining results are comparable without our cleavage treatment or with our cleavage treatment in advance for 24 hours. The staining results are quite comparable. So that results shows us our cleavage reactions don't damage the epitope in the cells. So with that two requirements fulfilled, then we do this really multi-cycle experiments. So on the top, what you are seeing is the conventional immunofluorescence results. So we are using fluorescent antibodies to stain different proteins in different cells. And on the bottom panel, what you are seeing is we are using different proteins and staining the same set of cells. You see that 1, 2, 3, 4, 1, 2, 3, 4 cells are always there. So on the bottom is the same set of cell. And to demonstrate the multi-cycle potential of our technology, in one cycle, we just stain one protein. So basically all the different protein images you see, we are staining them in different cycles. And on the bottom, we are using the same fluorophores. So as I demonstrated in the previous slides, we can stain at least 3 or 4 different proteins with different colored fluorophores in one cycle. But here, to demonstrate the multi-cycle potential, we are only using the same fluorophore to stain different proteins. So we stain different proteins in 12 different cycles using the same fluorophores. And as you can see, even after 10 cycles or 11 cycles, 12 cycles, our staining results is quite comparable to the conventional immunofluorescence. So obviously they are doing all the proteins in the very first cycle. And we are doing it after many, many cycles. Our staining results are also quite comparable. And we can further quantify those results. We can use our software to quantify the protein expression levels in individual cells. And we can overlay all the single colored image to give us this artificial color or pseudo color, the image. So we can visualize all the proteins simultaneously in one image. Then we can study protein-protein co-localization, their interactions, etc. And we can also study the single cell protein expressions using our software. And by comparing our results showing in red, even after 10, 11 cycles, our staining results is quite comparable with the conventional immunofluorescence. And this is another way to see the correlation between our results and the conventional immunofluorescence. It gives us very high correlation efficiencies. So this is the V2 assay Xiaoshan just mentioned. We are conjugating the fluorophores to the antibodies. And our goal to develop these technologies, we want to help the doctors or pathologists in the hospitals to diagnose the patient with much higher accuracies. Then later we apply our V1 technologies to the clinical samples. Usually they are or more than 95, more than 99% of the clinical samples in the archived tissue bank, they are formalin fixed, paraffin embedded tissues. We call it FFPE tissues. So when we apply our V1 technologies to FFPE tissues, we realize almost all the FFPE tissues, the clinical samples have extremely high autofluorescence signals. So when we are conjugating our fluorophores directly to the antibodies, the signal is too weak. So the signal is below the noise ratios, below the noise levels. So there's no way for us to visualize our real signals. So we ask the doctors, the pathologists, how do you stain your samples? So they tell us they are using these enzymatic signal amplifications. So they are using HRP, horseradish peroxidase, to deposit up to 100 fluorophores for one protein. So in this way, they can amplify the signals up to 2 orders of magnitude. So since obviously we have our cleavable linker, then we can insert our linker between the fluorophores and these pteramide moieties, and we can perform these signal amplifications using HRP. So this is the molecules we designed and synthesized, the fluorophores, pteramide moieties, and in the conventional TSA assays, people are using like this pteramide moieties to conjugate to the tyrosine residues on the proteins to stain the proteins. And with our cleavable linkers, so we can do the staining, and after the staining, we can cleave the fluorophores, and we discovered like simultaneously our cleavage reactions can deactivate HRPs. Then we can apply, after that, we can do imaging, then we can apply other antibodies labeled with HRP and deposit the fluorophores, then we do imaging, then we can, through cycles of staining, imaging, cleavage, HRP deactivation simultaneously, and cycles of staining, imaging, cleavage, staining, imaging, cleavage. They can quantify a large number of proteins with the high sensitivities. So first we demonstrate with this TSA-based approach, our signal amplification is two orders of magnitude higher than the conventional direct immunofluorescence or indirect immunofluorescence. This is our cleavable fluorescent pteramide technologies, the TSA-based approach. So comparing with the fluorophores directly conjugated on prime antibodies or the fluorophores directly conjugated on the secondary antibodies, our signals is about 2 orders of magnitude higher, and we didn't lose the spatial resolutions of our staining. So here that we are staining Cas67 in the HeLa cells. So this is one nucleus, and we can even see the nuclear line with our stainings. And with our HRP-based approach, we also can obtain the same staining resolutions. So all the staining resolutions is maintained, but the signal amplification, the signal intensities is amplified by 2 orders of magnitude. So only with these extremely high sensitivities, then we can apply our technologies to stain the biological, the patient biopsies. So this is the human hippocampus FFPE tissues, which has been sitting on the shelf of our collaborator for more than 1 decade. Then we apply our technologies, then we can stain different proteins on the these hippocampus tissues. So one spot you see basically is one nucleus. So first we use the new N to identify the neurons. Then we can, so here you see it's multicolor, but it's a false color. Basically, we are using the same fluorophores. To demonstrate the multi-cycle potential of our technology, we are using the same fluorophores to apply them in multi-cycles. So basically, all the images you see is obtained by Sci5, the same fluorophore. But here we are using the computers to assign them with the pseudo colors. And one spot you see is basically one nucleus. So we first identify all the neurons. We identify like 500,000 neurons in the same biopsies, in the same hippocampus tissues. Then we stain different proteins. Basically, all of them, they are transcription regulators in these tissues. Then based on the unique protein expression patterns, for the first time, we can classify individual neurons from the same patient biopsies into different clusters based on their unique protein expression patterns. Then we classify, I think around 500,000 individual neurons into 10 different clusters. And since we have the spatial resolutions, we can map them back to their original tissue locations. So we can see this is a dentate gyrus region. This is a CA4, CA1, 2, 3 regions. And we can see like in different CA regions and DG regions, the cell compositions, they are different. So in CA regions, we see a lot of these cell cluster 7 cells, but in DG regions, we don't see them. And even in the different part of the DG regions, we see the cell compositions is also different. So in this part, we can see visually like they have a lot of cells from cluster 2 neurons, but in other regions, we have a lot of the neurons are from the cluster 10 neurons. So what's the biology behind that? So this is the normal human hippocampus brains. So in Alzheimer's disease or in Parkinson's disease, in ALS, are we going to observe the same neuronal distribution patterns? Or specific neurons are selectively lost during the progression of the disease? And this is only the neurons. What about the microglia, oligodendrocytes, astrocytes, etc.? So during the progression of the disease, are they selectively activated? And with this spatial resolutions, we can study like is the specific neurons, are they surrounded by specific or activated astrocytes or microglia during the disease progressions. So this is something we are keep working on right now. And we can also apply the same technologies to quantify the tonsil tissues, to analyze the tonsil tissues. Again, this is the same fluorophores. So we just want to demonstrate the multi-cycle potential for technology. This is the same piece of tonsil tissues within the same Cy5 fluorophores. We can do many cycles. Then we can, based on the single cell, multiplexed protein expression levels, we can classify the cells from the same tissues into different cell clusters. Then we can map them back into the original tonsil locations. Then we can see like different locations in the tissues that are composed of cells from different clusters. So we can not only study the cell heterogeneities based on their unique protein expression levels. We can also study cell-cell interactions. So in this study, we classify the cells from the same tissue into 10 different clusters. And we can study which two cell subtypes they are more likely to associate with each other, shown in red in this picture, and which two cell subtypes they are more likely to avoid each other in these tissues. They are shown in blue. So we observe like cell cluster 5 and cell cluster 9. They are very likely to associate with each other. They are trying to be neighbors to each other in these tissues. And some cell clusters like cell 1 and 3, they are trying to avoid each other's. So they are always far away from each other in this piece of tissue. So what's the biology behind that? So later we can apply that to cancer immunotherapies. We can see like the engineered T cells or NK cells, are they close to a specific cell, cancer cell subtypes, or are they avoiding each other? Are they close or avoiding to each other? What's the microenvironment of the cancer cells determines the immunotherapy efficiencies. So this is something we are working on. And we can also further classify the cell clusters based on their neighbors. So initially, we define the cell clusters based on the protein expression levels. So all of them here is show all the cells here show they are cluster 1 cells based on their protein expression levels. However, we can further classify the cluster 1 cells based on their unique neighbors. So all of these are cluster 1 cells, but based on their neighbors, we can further classify them into a cluster 1A, 1B, 1C, 1D cells. So in this way, we can map them back, the cluster 1A, B, C, D cells, and we observe like different subclusters for cluster 1. They also have unique patterns on the tonsil tissues. So what's the biology behind that? So why, like based on their unique neighbors, unique cells have specific locations in the tissues, and how is that altered in diseases? So this is something we are keep working on right now. So basically, that's the story of proteins, and we can apply similar concept to profile RNA molecules in the tissues, in the specimens. So we can label the fluorophores to the oligonucleotides through a cleavable linker, then through cycles of staining, imaging, and cleavage. We can quantify a large number of different RNA molecules in the same specimen. So this is, again, our V1 technology. So here on the top panel, you see it's like a conventional RNA fish results. So they can stain different RNA molecules in different cells. And with our technology, again, with the same fluorophore, this is one cell, one HeLa cell. We can stain different RNA molecules in the same cell. So we can go from a low expression to high expression to low expression, low to high, high to low. And all of our results we obtained is highly comparable with the conventional immunofluorescence, conventional RNA fish experiments, even after many cycles. So this result shows like our cleavage reactions is really efficient and mild. So efficiency, we can go from high to low, low to high, and high to low. So it tells us like our cleavage reaction has extremely high efficiency. And even after many cycles, even the fragile RNA molecules are not damaged. Further confirms our cleavage reaction is extremely mild and efficient. again, when we after we develop this technology, we are very excited. But when we apply them to clinical samples, we don't see much. Like signals because of the extremely high autofluorescence signal in the tissues. So we have to again using the HRP molecules to further amplify the signals. So one HRP molecule enzyme can deposit up to 100 fluorophores. Only with this extremely efficient signal amplifications, then we can apply, we can visualize different RNA molecules in the clinical samples. So this is like one examples, like we can visualize different RNA molecules in the mouse spinal cord. So again, we are using the same fluorophores, and if we really zoom in, we can see like an individual spot, and one spot represents one mRNA molecules. And again, like we can classify the individual cells based on their unique RNA expression levels. Then we can classify the individual cells into different clusters. We can map them back into the original tissue locations. Then we can see like different regions of the spinal cord are composed of the cells from different clusters. So then we can further apply this technology to visualize a large number of different RNA molecules to study the disease mechanisms or to diagnose diseases with much more higher accuracies. And we can also apply this to visualize different DNA molecules using the similar technologies. Like again, like through cycles of staining imaging cleavage, we can visualize many DNA molecules through the same method. So this is one example. So in the same HeLa cell, this is the nucleus of a single HeLa cell. We can visualize the protein Cas7 expression levels. And later we can cleave the signals using the same fluorophores to stain mRNA Cas7s in the same cell. Then later we can cleave the signals and visualize the same genomic locations for Cas7s in the same cells. In this way, we can visualize the DNA, RNA, protein molecules simultaneously in the same cell, in the same specimen. And this is the clinical samples on the bottom. So people or pathologists have to stain different tissues, stain different biomarkers on different tissues. So on the bottom is the conventional immunohistochemistry or conventional RNA fish on the clinical samples. So they have to use different tissues to generate the same, to generate these multiplexed results. So on the top, so we are using the same tissue. You can see the same cells are there. On the top is the same tissue, and we are using the same fluorophores. We can stain the proteins. The first two are proteins. Then later we can stain the RNA molecules on the same tissues. So this is the protein RNA co-detections on the same tissues. So in this way, we can help the pathologist to diagnose the patient with much higher efficiencies. So to compare with some existing technologies on the protein side, so imaging mass spectrometry has been developed. And so the advantage of this technology is like they can apply all the antibodies simultaneously. However, they have to image the tissue sections pixel by pixels. So their imaging speed is a little bit slow. And so to image our tissue, usually like for most of the clinical samples, the tissues we are imaged is like centimeters by centimeters. And with our microscope, it's just the conventional epifluorescent microscope. Since our extremely high sensitivity, the signal intensities, we can reduce exposure time to just a couple of milliseconds. So to image like 1 centimeter by 1 centimeter tissue, our imaging time is 5 minutes. But to obtain the same resolution as we did to using this mass spectrometry imaging method to analyze the tissues pixel by pixel, we do the calculation for them. It may take a month, a couple of weeks to just the imaging time. So it's a little bit long for imaging if you are using the imaging mass spectrometry. And another technology, Codex, commercialized by Akoya, has been developed. They are using like fluorescent oligos to hybridize the oligo-conjugated antibodies. So it's similar to our V1 technologies. Per one antibodies, they can have maybe 2 to 4 fluorophores. So the signal is a little bit weak on the clinical samples, comparing with the high autofluorescence signals. And also, when we are developing the first V1 technologies, we discovered when we conjugated the fluorophores on antibodies, a lot of the antibodies stops working as good as before. So we have to validate a large number of different antibodies. So to develop a large panel of the fluorescent antibodies takes a long time. And with this oligo conjugated antibodies, so since oligo is a larger molecule, has a larger molecular weight than our small molecule linkers. So this one, I'm assuming is a little bit more challenging to develop the working antibodies. So it may takes a lot of time and efforts to validate the oligo conjugated antibodies are still working as good as the unconjugated antibodies. So with our approach, we can use the unconjugated antibodies. So this is a cartoon, but our approach, we can just use off-the-shelf prime antibodies directly apply them on the tissue samples. And our signal intensity is like 2 orders of magnitude than our V1 or maybe also the Codex technologies. So far, this is the advantage of our technology on the protein side. And on the RNA side, like other technologies like Moorfish, Seqfish, Cosmics, like we kind of developed them, the technology for them. And also the 10x genomics Sino technologies. So they are staining the same RNA molecules sequentially. So their advantage is like they have the extremely high multiplexing capacities. They can stain thousands of RNA molecules on the same specimen. So the disadvantage of them is like so far there's no way for them to apply the HRP signal amplification on them because there's no way for them to cleave the covalently linked fluorophores. So the signal intensity is relatively low for all of these technologies. So their detection sensitivity is a bit low. So most of their technologies in their publications, they don't claim they can apply that to FFPE tissues. And the third party do apply their technologies to the FFPE tissues. And they observe like the best performance among all of this technology, the best performance technologies can quantify. Usually, they're in the best scenarios, they can detect less than 0.25 copies for a specific gene in a single cell. So that's a little bit low compared to our HRP-based technologies. In our technologies, usually we can detect more than 5 or 20 copies based on the expression levels of the genes. But usually we can detect 5 to 20 copies per gene in individual cells using our technology when we apply them to FFPE tissues. So this extremely high sensitivity is our technology's advantage. And our low multiplexing capacity is kind of a limitation so far. So basically, we can develop, we can visualize up to maybe 100 different RNA molecules on the same specimen. So basically, we recommend our collaborators to do is like they can use like these technologies, high multiplexing technologies, to do the initial screenings. Once they identify their targets, maybe up to 100 different RNA molecules, then they can use our high sensitivities, high accuracy technologies to visualize these individual identified RNA targets in the clinical samples. Usually they are FFPE tissues. So with all of this technology we developed, then we can stain many different RNA molecules or protein molecules in the same tissues. Then we can study their signaling pathways, or we can visualize like which cells what's their microenvironment, which cell is associated with which cell subtype, which cell is avoiding other cell types, etc. And we can using these technologies to classify or to stratify patients into different groups. Then later we can give them a more effective therapies. So with that, I would like to thank all the funding resources and hardworking graduate students to work on these projects. And again, I would like to thank Sino Biological to give us this opportunity to introduce our research. And all the technologies we introduced here has been commercialized or will be commercialized very soon by Spatomics. And I will stop there and will be happy to answer any questions. Thank you very much.
Thank you very much, Dr. Guo and Zhao Shan, for your talk on cleavable fluorescent probe technology and its impact on the field of spatial imaging and, like you mentioned towards the end, patient stratification and its impact on diseases such as cancer. Our audience members today joined us from all over the world. We had members from Thailand, Brazil, Germany, India, as well as the US. It's always great to see where people are joining us from. We received a few questions throughout the presentation, and we can start addressing those. So Yuan Wang from Germany asked, would this technology offer the possibility to analyze transposon expression or mtDNA in the cytoplasm?
Yes, I think, yeah. So, when I'm giving this talk, like usually like among the audience, they will ask like, can you detect this or not? So my short answer to that is like, as long as any targets, they can be detected by conventional immunofluorescence, immunohistochemistry, or RNA fish, they can be detected by our technology. So our technology has no differences in terms of a probe design, etc. So all the antibodies working with the conventional approach, it will work with our approach. Any probes working with the conventional RNA fish, it will work with our approach. So the only difference is like we can chemically cleave the fluorophores, and we can apply other probes in the subsequent cycles to visualize all the different biomolecules simultaneously on the same specimen.
Great, thank you. Our next question comes from
I can
Hong
add a
Ling
little bit.
Xu.
Sorry, I can add a little
Yes,
bit
go
to the
ahead.
mitochondria DNA. So although there's a DNA, you could also consider using an RNA-H probe. The RNA-H probe, so although this RNA-H, but the probe is actually made of a DNA molecule. They are indifferent of RNA or DNA. You could design probe to target on the specific mitochondria DNA.
Thank you. Our next question comes from Hongling Zhu from the US. They ask, for this technique, when taking images at different times, is your software pipeline able to align the images automatically?
Yes, so far, yeah, our pipeline, yeah, in our publications, we already show like all the images can be aligned, and the Spatomics is working on that. So so far, they have made a significant progress. Like so far, I think our image alignment is the best on the market. So it takes a really short time and it's automatic. So we can do that. Yes.
Okay, and our last question from the audience comes from Karanvir Saini. They may have missed this in one of the slides, but they ask, using this technology, can we measure protein/RNA signal in live tissues? And what is a typical duration of each cycle?
So in live cells, so far, even for conventional immunofluorescence or conventional RNA fish, they are usually not applied in the live cell imagings because we have to get the antibodies, or we have to deliver the antibodies or the oligo probes into the cells. So usually we have to permeabilize the cellular membranes so to deliver these targets. Usually, like they are not applied in our in the conventional immunofluorescence or conventional RNA fish experiments. It's doable, but it's not widely adopted in the field. So and usually the cycle time, like in our lab, because we do everything manually before, so the cycle time is about 1 hour for proteins. So with the Spatomics, so they are building an instrument so they can further dramatically reduce the whole cycle time. And as I mentioned, the imaging time, our imaging time is just a couple of milliseconds to take one frame, and compare with the other technologies. They have the fluorophores and antibodies. Their imaging time is maybe on the seconds level. So our imaging time is reduced by 2 or even 3 orders of magnitude. So the imaging time is dramatically reduced. The antibody incubation time, we also have a way to dramatically reduce that with our engineered microfluidic system. So the whole assay time is further reduced. So it's quite fast and automatic.
Yeah, I want to add to that is yeah, so there's maybe the potential for live cells, but our current focus at Spatomics is on FFP tissue and also fresh frozen tissue because of the tissue availability.
Thank you. Since we have a couple minutes left, I will throw in one more question. We've talked about how CFP enables iterative cycles of staining, imaging, fluorophore cleaving. Could we talk a little bit more about the key benefits of this in regard to enhanced sensitivity or automation?
Sure. Yeah. In the field of spatial biology, almost all the technologies relies on signal staining and signal removal to stain other targets. Basically, it's a multi-cycle experiment. So in every cycle, they do the staining and they have to do signal removals. So people have been trying different methods to do signal removal. People have been using like strong detergent or very low pH, very high pH, strong light to deactivate the fluorophores. People or other companies, other technology has been developed. They are using different methods trying to remove the fluorescent signals. However, in our opinion, like other technologies, their signal removal method is quite harsh. No matter it is like a H2O2 based method, it can oxidize the epitopes, or the strong light, they can photo damage the epitopes, or low pH, or strong detergent like SDS. All of this method can damage the epitope to some levels, not mention RNAs. So RNA is more fragile. So to develop this cleavable fluorophores, so we have to make it our technology. So one of the major advantages is it is extremely mild. So this our cleavage reactions has been used in other biological assays for many, many years. It is extremely mild, and as we demonstrate, it doesn't damage the protein epitopes. They don't even damage the RNA molecules. So it is very mild, it is very efficient. So that's the major advantage compared with other methods. And so far in the market, like for signal amplifications, people have been trying to use oligo-based signal amplifications or the HRP-based signal amplifications. So for the oligo-based signal amplifications, usually the large complex has been formed, no matter it is the rolling circle amplification or the hairpin, the HRP, the HCR hybridization chain reaction based reactions. So this large biomolecule complex has been formed. So in FFPE tissues, so they are heavily fixed and are heavily cross-linked. It is quite challenging to form these large biological molecule complexes within the FFPE tissues. So the signal amplification efficiency may be limited for this large biomolecule complex amplification method. And with our HRP-based method, so our substrate is a small molecules, and they can penetrate very easily into the tissue sections very deeply. So it can give us very robust signal amplifications and very strong. And in our approach for RNA detections, we can even combine the oligo-based signal amplification combining with the HRP signal amplifications. So that's the reason like we can detect RNA molecules very robustly in the tissue samples. And in terms of automation, it's a small molecule fluorophores. And so far for the HRP-based method, so our method is so far the only method we can chemically remove the remove the fluorescent signals. So for other methods, there's no way to remove the TSA-based signal depositions. So that's basically the advantage of our technology.
The one thing to add is that the traditional technology using the TSA dye, as I mentioned during the introduction, that if you want to go to higher plexing, you probably can go up to 8 or 9, but a spectrum mixing capable scanner, fluorescent scanner, will be required to separate the spectrums. But for us, you can easily stain the 3 to 4 markers in each round, and after that, do the imaging, clear off the signal, and do another round. This gives a much less stress on the imaging system. You could use a regular fluorescent microscope or a regular fluorescent scanner.
Okay, as the hour concludes, I would like to once again thank both of our speakers, Jiao Shan and Dr. Guo. Great talk, very interesting to learn about your technology. We'd also like to thank both of you for using Sino biological products. It's been great to see your progress, and we wish you all the best. To our audience, thank you for joining us. And once again, to all those that would like to rewatch or share the presentation, it will be uploaded to YouTube later today. So with that, I will start to close down the webinar. Thank you both once again, and
Thank you, everybody.
good luck with everything.
Thank
Good
you,
luck
man.
at Spatomics and at ASU. All right, and on that note, we're signing off. Thank you very much to everyone who joined. Goodbye.